CIC lecture. So our lecture today is going to be given by Marina Broomfield. So Marina completed her bachelor's at McGill University in Pharmacology and Therapeutics, and it's completing her master's in the Computational Brain Anatomy Laboratory under the co-supervision of doctors Mallar Chakravarty and Rosemary Bagot. Her research focuses on exploring sex differences in healthy mice using neuroimaging and behavioral techniques, with hopes of improving our understanding of normative brain and behavior outcomes. So thank you, and
great. Okay, hi everyone. Can everyone hear me okay? Good. Okay, well, thank you for coming to my lecture today, where I will be talking about my master's project, assessing, can you see my mouse? Yeah, assessing brain structure and function in adult male and female mice using multimodal magnetic resonance imaging modalities and some behavioral analysis. Sex differences are prevalent across both men and women under healthy conditions in the brain. We can see these across both volumetric and functional measures, where, for example, males tend to have an overall larger brain size compared to females. And once we take this overall brain size into account, we see that there's still some significant areas where we see some sex differences. For example, females tend to have an overall larger prefrontal cortex, and males tend to have larger temporal and occipital areas. We can also see sex differences across brain function, where females tend to have greater activation in brain regions associated with language, which may then contribute to superior performances in verbal episodic memory. On the other hand, males tend to have enhanced activation in brain regions associated with navigation, which might give them an advantage during spatial reasoning tasks. Now, although we see sex differences under health, we can also see them under disease, where different prevalence rates are different across neurodegenerative and neuropsychiatric disorders. For example, we can see that males tend to have higher rates for autism, ADHD, and Parkinson's, and females tend to have higher rates for Alzheimer's disease, depression, and anxiety. In order to take these sex differences into account, in 2016, the NIH tried to encourage studies to include both sexes across their research. And although we do see that this increased over the 10 years, none of these studies actually really accounted for sex in an optimal way. What this indicates is that although most studies do include sex in their analysis, the way sex is assessed is not consistent or equal. We can also see this by the different proportions of male-only studies versus female-only studies. One thing to take into account is that this literature review was conducted including papers coming from cells, animals, and human work. And although the analysis of sex differences across human clinical trials have recently improved, animal models still remain behind. This is important to take into account because more often than not, we use animal models, especially mice, to test different types of therapeutic interventions, which we then test back on humans. However, for quite some time, there has been a historical male bias where only male mice have been included across research to test different therapeutics, which are then consumed across both men and women. This leads me to my research, which is to assess both male and female mice, especially in the C57 Black 6 mouse from the Jackson's laboratory. We chose this mouse strain because it's very popular across other research studies worldwide, and we wanted our study to be as generalizable as possible. The aim of my project, therefore, is to characterize behavioral, structural, and functional sex differences in the adult mouse. We do this by including a granular analysis of the open field test, as well as structural and functional magnetic resonance imaging. I'll now start by explaining why we included the open field test as a behavioral measure. The open field test is frequently used across many different research studies and measures anxiety-like behaviors. Anxiety is known to have higher prevalence rates across females. However, this test was actually developed to capture anxiety-like phenotypes in male mice. The way this test works is by once we grab the mouse from the cage, we place it in the corner of an arena, and then we let it investigate that arena for 5 minutes under red light. The outputs we get from this test are things such as the time spent in the center, times in the corners, the mean velocity, and the total distance traveled. However, by only looking at these behaviors, certain micro behaviors might go overlooked. These can be things such as rearing, grooming, moving forward, turning, and all of these micro behaviors are referred to as syllables. We can capture these micro behaviors by applying deep lab cut and key point MOISIC, which I will explain how they work shortly. But what's cool about this methodology is that it's all data-driven, so it avoids any type of experimental bias. We also included magnetic resonance imaging, since it's a non-invasive tool that allows us to study both animals and humans in a translatable fashion. In our study, we decided to focus on structural and functional MRI, where structural MRI can map the structure and morphology of the brain, allowing us to see whether there's certain anatomical areas that have higher volume across females or males, or lower anatomical areas. On the other hand, functional MRI measures changes in brain activity, which then induce a high metabolic demand, increasing blood flow to that specific area, changing the levels of deoxyhemoglobin, which are then captured by the MRI through the blood oxygen level dependent signal. Our study timeline is as follows. We first started with 40 mice, 20 per sex. And once they arrived from the Jackson's laboratory, we let them acclimatize for 2 weeks, after which at around age postnatal day 115, we exposed the mice to an open field test, followed by an MRI scan 24 to 48 hours afterwards. In order to increase our sample size, we then added 20 more mice, 10 per sex, which followed the same type of timeline. Okay. I'll first now start by explaining my behavioral data, going over the methods and then the results. We decided to include Deep Lab Cut, which is a software that allows us to label each body part of the mouse. And after applying Deep Lab Cut, we're able to use key point MOISIC, where we obtained 26 different syllables, each syllable depicting a micro behavior. Again, this can be grooming, rearing, moving forward. And after applying key point MOISIC, we obtained outputs such as syllable occurrence, syllable duration, and syllable transition frequency. I'll now explain what I mean by each one of these. When talking about syllable occurrence, we're talking about the amount of times each syllable shows up across the time frame of the behavior. So over here, what we see is in each color, a syllable, each syllable has a different color. Let's say syllable 18 is red. We see that syllable 18 shows up a total of 8 times across the behavioral test. When talking about syllable duration, we're now talking about the length of time that syllable was expressed for. In this case, it could be a total of 25 seconds. And when talking about syllable transition frequency, it's the amount of times syllable X is followed by syllable Y. So let's say syllable 18 is in red over here and syllable Y is in dark blue. This transition can happen a total of 4 times across the test. I'll now move forward to explain my results. First, throughout the normally reported open field measures, we see that females tended to have higher values for the total distance traveled and the mean velocity. And in order to further examine what these measures can tell us, we looked into our key point MOISIC outputs, where over here I have my 26 syllables on the x-axis, and each point represents 1 mouse for either my females in red or my males in blue. And across the syllable occurrence, we did not see any significant sex differences for any syllable. When looking at syllable duration, the same result appeared again, no significant differences in this either. However, when looking at syllable transition frequency, we see that we get some statistically significant results. Over here, I'm showing each syllable and its transition, where in red we see the T-stat for females and in blue the T-stat for males. And after correcting for multiple comparisons, we see that there's certain syllable transitions which are either higher for my females or higher for my males. This is a lot of data and maybe not the best visualization. So for this, Liz Herrera helped me generate the following plots. She's a PhD student at the lab who helped me a lot. But in order to better understand these outputs, we grouped each syllable into macro syllables. This can be behaviors such as grooming, jumping, rearing, turning, or moving forward. Once we grouped each one of these syllables into its macro syllable, we looked at the transition frequency, where now each edge is connecting two syllables, and the color of the edge is depicting whether this syllable transition happened more often across females in red or males in blue. What we saw over here it's still a lot of data, and I tried to come up with a way of okay, well, what does it mean to go from moving forward to then turning? But I'm scared of overgeneralizing things. So what I took from this was that overall, females exhibited more transitions than males. We can see this by the higher amount of red arrows than blue, indicating that females might have been more active during the arena. In conclusion, for our behavioral analysis, we included an open field test where we applied deep lab cut and key point Moisic and obtained significant sex differences across mean velocity, total distance traveled, and syllable transition frequency, with females having higher values for all of these, indicating a more active phenotype. We then asked ourselves whether there were any correlations between our behavior and our brain structure. And for this, we did partial least squares. The way this works is we have two different matrices, one containing our brain data, in this case, our relative Jacobians, and another matrix containing our behavioral data. In this case, I included all the normally reported open field measures, as well as the macro behaviors. We then correlate these two matrices, apply singular value decomposition, and obtain latent variables containing a brain pattern and a behavioral pattern, which covary together. In this result, we obtain one significant latent variable, explaining over 50% of the covariance. However, no significant sex differences were observed either across the brain loadings or the behavior loadings. Nonetheless, I was interested to see what this latent variable was telling me. Over here, we saw that this set of behaviors, for example, spending less time moving, more time being immobile, less time in the edges, these whole combination of behaviors correlated with either bigger or smaller volumes across the brain. For example, we can see this across the olfactory tubercle, the nucleus accumbens, and the caudoputamen. One thing to take into account again is that this latent variable did not exhibit any significant sex differences. In conclusion, in order to see if there were any brain behavior correlations, we applied partial least squares, where we did not obtain any sex-specific latent variables. Nonetheless, we moved forward and looked into our brain's MRIs. The way we did this is we applied a one-level deformation-based morphometry, which is an in-house pipeline developed by Dr. Gabriel Devigny. And after obtaining a structural MRI image for each one of our subjects, we iteratively use each subject to generate a population average from which we can then see whether a subject had to increase its anatomical region to match that of the population average, or if a subject had to decrease a specific anatomical area in order to match that of the population average. Once we applied this, we looked into our relative Jacobians, so taking overall brain size into account. To note is that after QCing, we had to drop 3 mice due to high rimming and hydrancephaly appearing in the scans. We also applied a linear mixed effect models where we looked at our relative Jacobians across sex with cohort as a random effect. And we then visualized whether areas were bigger across females over here in heat colors or smaller across females compared to males in cool colors. This is all showing an FDR 5% corrected threshold. And so we saw areas being either smaller for females across the olfactory bulb, the insula, and the amygdala, or areas being larger across females, for example, the motor, the somatosensory areas, and the thalamus. We wanted to further validate whether these results matched results recently published in the literature. This is a study published by Elisa Guma, where she looked at structural MRI scans of both humans and mice. But for the sake of my experiment, I tried to focus only on the ones from mice. And over here, she's showing structural images of the unthresholded brain maps and the significantly thresholded maps. Through this, we were able to see certain areas were similar across our studies. For example, the olfactory bulb, the prefrontal cortex, and the striatum. However, there were certain dissimilarities, which could have been due to the fact that her sample size included mice that were half the age of mine, and she also included different strains of mice. In conclusion, what we can take from our structural data is we applied deformation-based morphometry to obtain relative Jacobians, from which we saw that females tended to have an overall larger brain size compared to males. And we saw that males tended to have larger brain areas for the olfactory bulb, the amygdala, and the insula. We then moved forward with our functional MRI analysis, where we had our mice anesthetized with an optimized protocol developed by an old master student at the lab, Mila Jurosevic. Initially, we anesthetized the mice with 3.5% isoflurane, and we gave them an IP bolus injection of dexmedetomidine. And while they were getting scanned, we gradually decreased the amount of isoflurane they were exposed to until it reached 0.2% by the time that they were getting the functional MRI acquired. I also used rabies, which is a pipeline developed by a PhD student at the lab, Gabriel Desrosiers. This pipeline allowed me to quality control and to analyze my functional images further. We applied group ICA and dual regression to our functional scans. The way group ICA works is it generates a matrix for each one of our subjects having voxels across time, from which the synchronous activity across many voxels consistently showing up throughout the time series for all of our subjects get mapped into each network. And once we have the network time series, we map them back into the spatial map to see where they are located. Once we acquire these group ICA spatial maps, we use them to obtain the subject-specific time course during our dual regression step, from which we can then grab that time course and generate our subject-specific spatial maps. After applying group ICA and dual regression, we obtained 4 different networks, one across the somatomotor, somatosensory, thalamus, and the striatum. And after QCing for our scans for any high movement or mice that might have been overly anesthetized, we ended up with 18 males and 23 females. What we saw over here was that after correcting for multiple comparisons, females tended to have within network amplitude higher for the somatomotor and somatosensory networks. We then wanted to see whether there was any functional connectivity across these networks. And for this, Dr. Johan Yi helped me generate the following plots, where over here I'm showing the Fisher set transformed correlation values for the upper triangle, either for my females, and the lower triangle for my males. And in order to see whether these correlation values were statistically significant, statistically different across sex, we have this plot over here showing the T statistic in red for my females and in blue for my males, where we see that males tend to have higher functional connectivity across the thalamus and the somatomotor networks, as well as the thalamus and the somatosensory networks compared to females. And this is after applying multiple comparison correction. This was very interesting to us because there has recently been a paper published with rats where they assessed resting state functional connectivity across both males and females. And what these circles are depicting are areas across the brain forming parts of bigger regions across the brain over here with the legend. And what they concluded from this study was that males tended to have higher between network connectivity compared to females, whereas females had higher within network connectivity. This was also seen throughout the cortex and the subcortex, where males tended to have higher functional connectivity connecting both the cortex and the subcortex, and females tended to have more segregated activity across both. Another study was also looking into functional connectivity in humans, where they also reported similar findings, where we can see males had higher between module connectivity and females had higher within module connectivity in teenagers. This then led us to our next question, which was, does this also apply in mice? Do we see this segregation across the cortex and the subcortex? And for this, we applied seed-based connectivity, where the way this works is after acquiring our resting state functional MRI, we generate regions of interest, in my case, located both across the cortex and the subcortex. And we then examine their time series. By examining their time series, we can see whether these regions correlate with each other or if they don't. So if one of them is active, what is the other one doing? Is it also activating at the same time, or is it being suppressed? After looking at this, we can generate a correlation matrix, allowing us to see this. In my study, I included 12 different seeds, and I'm going to zoom into one of these plots in order for you guys to better see it. Over here, I'm showing a seed I generated for the primary sensory cortex, outlined in green. And what we're seeing are brain regions which are correlated with the time series across this seed. In this case, we can see that after correcting for multiple comparisons, females tended to have an overall higher correlation across the brain for the primary sensory cortex. This trend showed up across other areas, such as the primary motor cortex, primary sensory, and primary secondary cortex, frontal association cortex, and the thalamus. Males, on the other hand, tended to have higher functional connectivity for the infralimbic cingulate cortex and other brain regions. Similar to what we did with our dual regression results, we wanted to see the correlation values across each seed. So what happens when the dorsal striatum is activated? Is the frontal association cortex also activated or not? And in the upper triangle, I'm showing values for my females, and in the lower triangle, values for my males. In order to compare these, I'm showing over here the T statistic, where we saw this reoccurring trend showing females had higher functional connectivity between the thalamus and the dorsal striatum, and between the secondary motor cortex and the frontal association cortex, again, segregating the cortex and the subcortex differently. However, males tended to have higher functional connectivity linking both the thalamus and the insula. Once we obtained these results, we applied a more rigorous analysis, a statistical analysis, where we applied multiple comparisons, and we did not see this effect any longer being statistically significant. In summary, what we did with our functional MRI is we applied group ICA and dual regression, followed by seed-based connectivity, where we were able to see that females tended to have higher within network amplitude for the somatomotor and somatosensory cortex, and males tended to have this trend of having higher intercortical connectivity across the cortex and the subcortex. We then asked ourselves whether these correlations, whether there were any brain behavior correlations across brain function and behavior. And for this, we applied partial least squares. In my PLS, I included, again, the normally reported open field measures along with the macro behaviors. And for my functional MRI scans, I included the dual regression time series results. Here, we obtained one significant latent variable, explaining close to 50% of the covariance, where we saw that being female, spending more time in the center, less time in the edges, more time moving forward, and going from grooming to jumping more often, positively correlated with functional connectivity between the striatum and the somatomotor, and negatively correlated with functional connectivity across the thalamus and the somatomotor. This was very interesting because at the beginning of the presentation, I showed you guys females did tend to be more active during the open field test. And during our functional connectivity results, we also saw that males tended to have higher functional connectivity between the thalamus and the somatomotor and somatosensory cortices. So I'm going to give you guys a brief summary of what I've presented so far. In our open field behavior, we saw that, again, females were more active. In our structural data, we saw that males tended to have an overall larger olfactory bulb, amygdala, and insula. And females had larger amounts for several regions, such as the somatomotor, somatosensory, prefrontal cortex, thalamus, and the striatum. Across our functional results, we saw that females had higher within network amplitude for the somatomotor and somatosensory cortices. And we saw that males tended to have this higher cortical and subcortical functional connectivity. For our PLS, we only saw significant sex differences across our brain function and behavior, where being female, being more active in the arena, correlated with weaker functional connectivity across the thalamus and the somatomotor, and stronger functional connectivity between the striatum and the somatomotor. So what can we take from these results? There are sex differences in the C57 mouse, and these are very important to consider before moving on to disease models in order for you to be able to see what the effect of the drug you're using or the disease you're using are on the model you're testing. And probably the most important slide of my presentation, I wouldn't be able to have done this without the help of my amazing supervisor, Mallar Chakravarty, and my co-supervisor, Rosemary Bagot, as well as with the help of Liz and all of my lab members. You guys have been amazing. And my mice, because they deserve a shout out.
Oh, I just clicked again. Is it on now? Okay. Okay, so thank you very much. That was a really good talk. Is there any questions from the audience, either here or online? We can start with Khalil and then Qatar. Let me give you the mic.
Hi, thank you so much. It was a wonderful work. It was really, really good. I just was wondering about the obvious, about the estrous cycle, if you check it, or how you think it could be a difference in your results.
We did not check for the estrous cycle. The experiment was conducted in like 3 days per each mouse. And the estrous cycle, at least in human, varies across a bigger span than that. Males do tend to have higher fluctuations across their hormones on a daily basis, which actually creates more variation across my male data than my female data. But I mean, a study where they do take that into account would come in handy for sure. Yeah.
Thank you, Marina. That was a very nice presentation. I guess it's my question is a bit related to Khalil's question, but I'm curious to know what's the theory behind sex differences in brain development. Like, is it related to hormonal surges during development, or is it something else? What is your idea about this?
In a paper where they were looking at structural MRI, they were they were seeing that there were certain brain regions that developed sooner across males compared to females, and then they remained bigger in males than females, which kind of leads me to think that maybe males have this like accelerated brain growth for certain regions compared to females, that just develop at different rates. But for our functional MRI, usually across humans, the prefrontal cortex develops way sooner across females than males, leading to more like rational thinking while you're like in a teenager phase. But there's for sure developmental effects that happen. This is why we focused on adult mice. So we hope that by the time they do reach that age, those effects are subsided.
Kind of normalized.
Yeah.
Awesome. Thank you.
Is there any more questions from?
In your opinion, what is like the optimal sex design for studies using these mice in the future? Like, do we incorporate males and females in one run analysis and kind of like look at sex after? Do we run like a sex stratified analysis? Like, what in your opinion is like the way to go?
I would compare them differently. I would run an analysis across males and then an analysis across females. If you don't see any different results, go ahead and combine them. It'll give you more power, but by combining them right away, you might actually be overlooking effects that might be driving certain results that are just hidden because you don't see it.
Anyone else here?
I'm going to push back. Do you think that's actually true?
On the
Why do you think that's true? Why do you think that if you combine your animals right away, that you would obscure results? I mean, both things are possible, right? I mean, what would combining all your animals at the outset tell you? And then what would doing a sex-specific analysis tell you? Both are equally as valid, but they just tell you different things.
But wouldn't you think that there might be like okay, let me
Don't
give
ask
you
my
an
question with a
example.
question.
Let me give you an example.
I'm using my privilege here to ask.
Yes, fair enough. Let's say, for example, you're looking at the development of the prefrontal cortex, okay?
Yeah.
And you're combining both sexes from the get-go.
Yes.
You might think that this development is actually accelerated compared to other brain regions. However, it might only be the females driving that result compared to the males.
That's not how your statistics work.
Well, it would average it out.
Okay, so what would you learn from that then?
So then you would lose the information that
No,
is
you wouldn't.
developing across.
You've learned something by doing that. What did you learn? If you don't have a result, doesn't mean you didn't learn anything. So you include equal number of males and equal number of females in the same experiment, and you get a null result. What did you learn about that phenotype?
That combining males and females in an experiment won't give you.
Well, that by having a population-based model that includes males and females,
Yes.
you don't have an effect, right?
Right.
Then if you go ahead and then disaggregate the data and put it into a male-specific model and a female-specific, you can then uncover why that's true. But both are valid answers.
Yeah.
Right. And both are necessary analyses that need to be done to actually better understand what's happening.
Yes, I agree.
Okay.
But let's say, why wouldn't you do it from the get-go, separating males and females? You might actually see the result before.
What's the goal?
Rather than.
Is the goal to find out what's happening at a population level? Or it depends on the goal. If the goal is to figure out what's happening at a population level,
yes,
then no, you necessarily need to combine everyone, right? If the goal is to figure out what's happening in males and what's happening in females, then it doesn't matter what's happening at the population level, right? Right. I think
yes.
I think these blanket statements are important, but they actually may do more harm than good, because you actually, I'm all, as you know, I'm all for. I think we need to do a much better job with including females in our analysis, but that doesn't preclude the idea of that there's a scientifically, there's a population that you've sampled that you need to further examine. We can disagree. You can feel free to disagree.
I would tackle it with sex differences first and then combining the population. But I do
Same
think
difference.
both
It's
are
just like two
important.
sides of the coin, right? But yeah. I just would throw out one for the other. That's all.
Yes, okay.
Anyone else? Yep.
I was thinking about just the developmental trajectory and which point, which age is like the females and males are separate. Like this difference is maybe you can find any results if you check the mice at I don't know 10 days of after birth, and but maybe in some point, maybe in 20 days or maybe 30 days, the hormones makes the job, so it starts to separate the difference. Do you think, or there is something in the literature that tells something like that, that some point the hormones are starting to increasing some differences?
Like at some point the hormones start
Yeah.
to spike and
Yeah,
then
yeah, yeah.
they enter like puberty, I guess, and
Yeah,
all
like
of
like
these
these
different
stages in the human developmental or in the mice developmental, there is like a
Yes.
big change that you can measure, because at the end we are measuring it in the macro scale, not in the just micro. Maybe all these changes are starting from micro, and then at some point separate in the macrostructure. We can see it.
And I do think that is the case. I think there's, well, we're developing usually until the age of puberty. There are certain differences across the brain. However, we see a big surge of neuronal differences once puberty starts and hormones do start getting involved across the brain and affecting different brain regions like the amygdala. Yes.
Thank you.
Thank you for your talk, Marina. Very nice presentation. I was wondering why, if you have like a hypothesis or know why there might be more intra-network connectivity for females on the somatomotor side. Like in your other behavioral experiments, you also saw that they were like more active. Is there like a reason? Because I would think that males would be more active. I don't know.
That's a good question. That's a really good question. And I wish I had a great answer for it, but I don't. It's still like, I'm still trying to wrap my mind around it. The open field test has been developed to measure anxiety-like phenotypes in males. So it's hard to interpret the outcomes we get in a female perspective, right? So what might be spending time in the corners, which is associated with more stress, might be the case for male mice, but might not be the case for females. And so being more active in the arena, you would think, oh, maybe they're less stressed, they're moving around more, they're spending more time in the center. However, what I don't know if that actually tells us whether they are stressed or not. More research has to be done in that aspect. And towards my functional results, could you repeat that question? Sorry.
Yeah, it was around the same, like why intra-network, why intra-network.
Okay, I see. Why do we see that segregation?
Yeah.
I don't know.
First, I want to compliment your presentation, specifically the clarity of your presentation, because like I was able to understood everything from the beginning to the end, so it was very good. And yeah, just the end of it, like the conclusion that, hey, there's actually differences between females and males. I thought maybe you could just expand on that, like saying, because you mentioned like certain areas are different, certain connections are different. So maybe because at the end you said like you can split, you should split your data into two, or like when you're handling your data, maybe this can be more specific, like if you're targeting specific areas, if your drug is like targeting specific connections, if so and so, then you need to split your data. If not, then you can like have more data, just to like extend.
Okay, I agree. Yes, it's very well said. Yes.
Anyone else? So I have one. So in your PLS analysis, where you show like you grouped your behavior syllables into macro behaviors. So let's say that you have different syllables of grooming and you group them all together. Are you referring that all grooming is the same, or is this driven by a specific syllable of grooming?
So we actually ran the PLS analysis also across each individual syllable instead of grouping them all. I have the results in my supplementary over here. And what we see is a very similar trend, but flipped. So now for my male subjects, spending less time in the center, more time in the middle, spending less time moving forward, less turns, spending more time rearing and moving forward, and then less time going from forward to rearing, gives me the opposite brain pattern than I saw. So I see that sex is an effect, and like I decided to show the other ones beforehand, just because of the simplicity of reading the behavioral loadings, just to get a better representation. But we did look into the syllable specific effect, and whether we group the syllables or not, they're more or less telling us the same thing across sex.
Okay. Anyone else? There's no questions online, I guess. Okay, well, so thank you very much, Marina. It was a great talk. And enjoy the rest of your afternoon.